poster session in the sense we have a lot of posters, I believe 24, so there are a lot of people presenting. They all have 2 minutes. And we understand this has nothing to do with science. This has everything to do with advertising. So the posters are out there, the science will be there. You are allowed to advertise your poster, so do that as good as possible. We have a very rigid schedule, there's no way around it, otherwise we cannot manage it. So I have a timer, and the timer I even have some personnel for the timer, that is Tim Pedley. And since it is his kitchen clock, he knows to handle that best. And it's also good to uncouple the chairmanship from the timing, so Tim he just starts, and 2 minutes, the next 2 minutes starts. And the same with the pointer I have of the slide processor that I have here. So if you are not rapid enough, you may talk in front of a slide from the next speaker. So please be in time. So that's enough. You can line up there, so let's not lose time by switching speakers. So if one speaker is busy, then another one is standing there in the front, so then we can really manage this through. Okay, may I have the first one? Jose Sanz. This is a test of me as speaker of all those names from different countries. Is the first speaker there?
Yes.
Okay, please go ahead.
Thank you. Okay, my name is Jose Francisco Sáenz. I am a PhD student at the Department of Biophysical and Electronic Engineering at the University of Genoa, Italy. I wish to present you this poster in which I am proposing using a platform that combines the atomic force microscopy and microelectroarrays for studying the cardiac mechanoelectric feedback phenomenon. So the motivation is arrived from the well-known fact that the mechanical state of the heart alters the electrical activity, not only in physiological way, but also in a pathological manner, mechanically induced arrhythmias. So in the platform, with the FM, I am able to stimulate the tissue, the cardiac tissue, by applying local deformation, controlling the force, timing with the spontaneous activity, and also at the same time providing multi-point electrophysiological recording. So I also considering in my work to use computational modeling for predicting and help to understand the extracellular signal that I am recording. So this is an example of the signals of the preliminary experiment I have done until now. And I can see that the spontaneous response depends, the morphology depends of the timing with the spontaneous activity. And there is several analyses that I am performing, and also I am trying to help with a computer model that simulates a strand of cardiac cells and the recording with several recording electrodes, starting from the intracellular signal, trying to predict the extracellular signal I am recording. So I still have a lot of work to do, but with the preliminary results, I think that this proposed platform can help for new studies. So thank you. If you are interested, come to my post.
Good, thank you very much. So the next speaker is Michael Moulton.
In treating patients with heart disease, I'm often struck by our lack of the understanding of the basic pathophysiology of many of these diseases. An example would be diastolic heart failure, where apparently there are millions of people who have this disease, perhaps hundreds of thousands that die each year, but we really don't understand the basic muscle or tissue defect underlying this disease. Certainly we have the capability of obtaining very detailed measurements of these patients' hearts in vivo using echo or MRI, but what we lack is a consistent modeling framework with which to synthesize this data. Partially to rectify this problem, we've developed this mathematical model of the heart, which starts with a very simple geometric assumption. The heart is represented as a thin-walled cylinder. Helical muscle fibers are embedded. We impose displacement boundary conditions on rotation and translation, but we allow torsion of the muscle fibers. We provide realistic active and passive muscle properties. breaking with other models, we don't prescribe display or pressure boundary conditions, but allow the pressure to be a variable in the simulation. When you add together the equations that describe stress equilibrium with the equations that provide some geometric constraints, as well as those that allow interaction between the pumping heart and the afterload and the preload, you get a system of differential algebraic equations which are solved. These are time-dependent equations. They're solved using a backward differentiation scheme. And this yields a series of realistic pressure curves, pressure-volume relationships, strains that span multiple cardiac cycles. And it's our hope that we can take this model and couple it with patient-specific data to understand the basic muscle properties in diseases like diastolic heart failure.
Thank you. Move on to the next speaker, Martin Kruger.
All right. My name's Martin Kruger. I'm from the Institute of Biomedical Engineering of the Karlsruhe Institute of Technology, and I'm concerned with the human atria. Like my fourth speaker said, for him the ventricles are like a cylinder. Well, I'm kind of like concerned what's on top of the cylinder. So the human atria consist actually of thin atrial walls, but within these atrial walls we got fine myocardial structures, and those can't be seen on image data. Among these are the Bachmann's bundle, the terminal crest, and the pectinate muscles. And these structures have a very fast conduction velocity, and they influence the excitation sequence of the atria very strongly. So if we want to build patient-specific models of the atria, we certainly have to consider those structures. So what I was doing is I was translating image data onto geometric homogeneous models of the atria, and then enhancing these models using a semi-automated algorithm with the fine myocardial atrial structures, and then conducting simulations with those models and compare the excitation sequence as well as the activation times of those to see which structures really need to be included into patient-specific atrial models. So we have one example of our results here, which shows that the Bachmann's bundle has its greatest influence on the left atrial appendage from the activation time. And if you look at the overall activation time of the left atria, it will be decreased by about 15% just from the Bachmann's bundle. So if you're interested in atrial modeling and anatomical and electrophysiological modeling, please come to my poster. It's number 3, and talk to me.
Okay, thank you very much. Next speaker, Osama Yarous. Go ahead. Title is not there. I don't need to read.
Well, hello everyone. I'm Osama. Please don't be panic. I'm a PhD student at the University of Karlsruhe, and my PhD thesis is about efficient, personalized, elastomechanical modeling of the heart. And I'm using a mass spring system, a modified mass spring system, for that reason, that addresses the problems that we have in elastomechanical modeling, the anisotropy, the nonlinearity, and the volume preservation. So this is an old snapshot, but it shows a truncated ellipsoid as a model of the left ventricle. And you can see a fiber twist, you can, an apex twist, and you can also see, the walls are getting thicker. If you want to see a better snapshots, you can visit my poster. It's number 4. So thank you.
Great. Good example. Yasmin.
Hello, my name is Jasmine Aguado Sierra, and this is some of the work that I've been doing at University of California in San Diego. So if you visit my poster, you will see some of the results of a model of electrophysiology I'm using a simplified ventricular model of a dog. and introducing cell heterogeneity. But not only transmural heterogeneity, but also apex to base heterogeneity. That's why, well, we've all seen today that the myocardium is very heterogeneous. So some of the methods that I've implemented in this model, it is a simplified model with 5,280 elements, 30 elements transmurally. I am using the model developed by Sarah Flame, that it's a modified Greenstein model. Professor McCulloch talked about it a bit yesterday. And it is a model that includes ion channel heterogeneity and also calcium handling protein heterogeneity. So I am also using some orthotropic conductivity following the data by Caldwell. I'm using some apex to base fiber and sheet angle orientation heterogeneity following some data by Costa. And also I'm introducing some gradual or gradient of heterogeneity of ITO and IKS to simulate the differences in action potential duration between apex sites and basal sites. So these are some of the results where you can see that actual, perhaps, depolarization of the whole ventricle is very similar in apex to base, but where we can see a difference is on repolarization.
Okay, yes. You could finish that sentence at least. So. Next speaker, Aaron Holden. That's 20 seconds lost coming from your chair. Oh, I will hear. So this is your good.
Okay, I'm Aaron Holden from Leeds. For the past 20 or so years, I've been looking at reaction diffusion equation models of cardiac tissue. I've been concentrating on the reaction term. I'm now looking at the diffusion term, how things spread with distance, and we're using diffusion tensor imaging to get data. And the kind of assumption of reaction diffusion modeling is we can explain wave breaks into tachycardia, into fibrillation. This is the kind of reaction diffusion model, or Untitled medium model. If you look at the heart, it doesn't look like a continuous medium. It's got holes in it, it's got vessels, it's got cells. And so my solution to that is to make available publicly through a 48 terabyte data, all our data. This is raw MRI data, diffusion tensor image MRI, and everything else, and allow people to look at it. And this is for a variety of animals, ranging from rats, people, pigs, to rabbits, you know. So it is available. This facility, if we were to populate it with our data, it would take us 100 years to fill it. So we're also inviting other people to contribute data to this. But the belief is we're publicly funded science, so it should be publicly available, and we're not worried about IPR. So anything on there is for anyone to use. Thank you.
Thank you very much. Let's bring it up. So, Kenneth Tran.
Good afternoon, or good morning, sorry. My name is Kenneth and I'm from the Auckland Bioengineering Institute, and our study here is concerned with trying to understand the effects of ischemia on cell function. Now, during ischemia, a mismatch between energy supply and energy demand leads to a dysfunction in how the cell can contract. So what we've done with our models is that to understand ischemia, we need to be able to model the effect of changing metabolites on the three main ATPases in the myocyte. What we've got here is the circuit pump and cross-bridge kinetics. In this figure here, under the circuit pump, what it shows is that when the net free energy becomes positive, the circuit pump actually can function in the reverse direction. For our cross-bridge model, as a validation of it, this figure here shows the effect of a sinusoidal length perturbation on the complex stiffness at different inorganic phosphate concentrations. And we can see that it matches up quite well with the data from Kauai. With our ATPase models, we then couple it to Danby's mitochondrial model, and this is coupled into a whole cell framework, which includes electrophysiology, EC coupling, and calcium dynamics. And we use this integrated model to try and analyze the relative contributions of each of these three ATPases to the total energy consumption at different work rates. So I think I'll just stop there, and if you're interested in the results of our simulation, please come and have a look at poster 7.
Good. Sinds je niet Tim af. Oh, hier. It's good. Look from here.
Yep. This is not my sheet. Thank you. I'm Joep van Leer. I'm from the Technical University in Eindhoven, where I'm currently doing a PhD. And the poster is on some work I did at the Medical College of Wisconsin in collaboration with Daniel Beard. We are interested in biochemical networks, such as the one shown in the top right, where we want to take into account proton and metal ion buffering, multiple compartments, and we want to have as many constraints as possible. And one of these is that it should be thermodynamically feasible, which means that there should be no flux in thermodynamic equilibrium. Furthermore, the thermodynamic equilibrium is affected by pH, ion binding, and electrophysiology, such as magnetic potential. To this end, we've made a framework which enables us to construct these models, because building such models by hand is quite error-prone, where we have a database of thermodynamic constants, which are used to compute the thermodynamic constraints. We have component models of enzymes and transporters, which are based on isolated enzyme and transporter studies. We have a database of metal ion ligand stability constants, which are used to compute ion binding and proton binding to different reaction species. And we have a BSL file which specifies different compartments and enzymes which are active in different compartments and transport between different compartments and their associated models. So then we can run the builder, which gives us the right-hand side of our large-scale model, which we can subsequently use to do full system studies. So I think I'll conclude with that. Thank you.
Thank you very much. Next speaker, Terkilzen, please.
Hi, as I've heard, my name's Yona. I'm from Auckland Bioengineering Institute. As I've heard a couple of times in the past couple of days, alternance is a beat-to-beat alternation in both the action potential duration and the amplitude of calcium transient. Quite often, alternance is studied in the context of rapid pacing. So when you increase the pacing frequency, then alternance will begin. But what you also see is that during ischemia, alternance develops within the first couple of minutes, or not always, but during severe ischemia, it will develop during the first couple of minutes of ischemia. And so what my poster is about is the development of alternance during a model of zero-flow global ischemia. And what we've found is that we think there are two mechanisms which are acting to cause the alternance, one being the well-documented calcium overload mechanism, and the other is slightly counterintuitive, which is, we think, associated with the opening of the ATP-dependent potassium channel. But we need to work a bit more on this and talk to some people, and so if you are interested in this kind of work and would like to talk to me about it, please come past. I think I'm poster number 9. And yeah, good to see.
Okay, thank you. Next speaker, Schmitz.
Hello, my name is Joep Schmitz, and for my poster we changed from cardiac to skeletal muscle. And as Jeroen Jennissen explained in his talk, is that we can use skeletal muscle, its unique properties, to study metabolism and develop models that can also be useful for cardiac modeling. So what we did was we developed an experimental setup to record phosphoramidol spectroscopy during bicycle exercise, and we used this data to test and validate computational models. So we constructed a cell model of skeletal muscle. We included the model, the Beard model of mitochondria, and the Lambert and Kishmarek model of glycolysis. And one of the properties of a skeletal muscle is one of the multi-scale aspects is that it contains different cell types. So we also developed what we call organ model, which includes those different cell types. And we use these two methods and tools to analyze the deactivation of glycolysis in non-contracting skeletal muscle, which was briefly mentioned by Jeroen on Monday. We also studied the magnitude and control of mitochondrial ADP sensitivity, which also explained by Jeroen. So if you want some more details about that study, it's on the poster. And we also predicted the individual fiber type behavior, so the dynamics of the metabolites and individual muscle fibers. So if you're interested, please come and visit the poster. Thank you.
Thank you very much. So, next speaker.
That's true. Well, my name is Willemijn, and I'm also working on skeletal muscle cells, but on the spatial temporal behavior. Because what we noticed was that healthy muscle is highly organized, but that in diseased muscle, like type 2 diabetes, but also in certain knockout mice, this structure can completely change. Well, how did we attack this problem? We developed a spatiotemporal model because some of these parts we cannot measure. But we also did some experiments to set some criteria for our model. So what we did is we performed calcium measurements, but we also performed some separate force measurements to be able to verify the model. Well, this is the spatiotemporal model. First we have the calcium model, it's a sarcomere model. Then we included force prediction based on the cycling cross-bridge theory. But now we're also working on incorporating the energy metabolism that Joep just taught us into this model. Because as you can see, the mitochondria also have a fixed position in space. Well, with these methods, we've been able to show that the local calcium concentration can differ from the spatially average concentration. We have identified which are the most important regulatory mechanisms in force development, and we also showed that there are also gradients in energy metabolites. Okay, thank you.
Thank you. My poster is really about the way that structural data gets incorporated into computational models when the data is obtained from images at a range of resolutions and really looking at tissue electrophysiology. And the background to it is that we can acquire structural data at a whole range of scales, from confocal at submicron to micron, histology at microns to tens of microns, and MRI at tens to hundreds of microns. And we analyse this data, get structural orientation or detail, and then look to put that into a computational model which may be at a completely different resolution again. And some of these issues were apparent in some of the talks this morning. So there's really just three short snapshots on the poster. One is the effect of scale. It's hardly surprising to realise that the resolution at which you acquire your structural information and use it in your solution makes a difference to the results of that solution. But this effect can be quite large for surprisingly small levels of change of resolution. So one part looks at that. Second part looks at obtaining information about structural orientation in a way that lets us create a hierarchy of scales of representing that structure. And the third part is to do with modelling structure based on experimental work and incorporating various levels of detail from different imaging modalities into a computational model to replicate the experiment, and which relates very much to what Vicente said this morning. And it seems that no matter how much detail is in there, you still are only approximating what you record in an experiment. And the rest of the information's on the poster. Thanks.
Thank you very much. You see, the amount of applause is proportional to the shortness. So the next speaker, Adelaide Devecchi.
So good morning, my name is Adelaide Devecchi, and today I'll show you some of the work I've been doing at the Computing Laboratory in Oxford on fluid-structure interaction in the congenital heart. So the starting point for this work is a systemic right ventricle from a 3-year-old baby with a condition called congenitally correct intransposition of great arteries. So for those of you who are not familiar with this terminology, this condition involves a double discordance, so atrioventricular discordance and arterioventricular discordance. This means that for this child, oxygenated blood goes from the left atrium through the tricuspid valve inside the right ventricle, and from the right ventricle is pumped to the aorta and delivered to the body. So the systemic circulation is supported by the right ventricle, and this is compatible with life, hence the term congenitally corrected transposition. But of course, it's not ideal. So these patients have like a short lifespan, and they have a series of problems, including heart block and heart failure. So this gives us the motivation for this study. We're trying to understand what's going on in the right ventricle when it copes with the high pressure due to the systemic circulation. And from this, determine the clinical metrics for this group of patients. So I've coupled the solid mechanic model with the fluid mesh, and I've run a simulation of diastolic filling using this inflow velocity profile. And this is a movie that I got of the vortex formation and the solid displacement. And if you're interested in knowing more about this movie, my poster is number 13. Thank you.
Next speaker, Oleg Aslanidi.
That's fine, thank you. All right, thanks. Oleg Aslanidi, Manchester. I'll speak about atrial fibrillation. AF is the most common sustained cardiac arrhythmia, but its mechanisms aren't well understood. From clinical data, it's known that in the right atrium, activation pattern is quite similar to that in VF, with multiple wavelets and quasi-chaotic activity. The most interesting.
Go ahead. No.
Thanks. The most interesting thing happens in the left atrium. You can see it at the top left. There is a single source which sends periodic waves, and its high frequency it dominates overall activation pattern during AF. Same is seen in both clinical data and isolated tissue experiments. The only apparent difference between right and left atria is electrophysiological. It's a small difference in action potential duration, which is also translates into small difference in effective refractory period. We constructed geometrically simple tissue model for both atria, which takes into account detailed electrophysiological differences between them, right and left atria, and also several intraatrial conductive bundles. And in such a system, if we start from a symmetric initial condition, which corresponds to two rotating spiral waves, we can see that symmetry breaks down pretty rapidly, and within few seconds, we get activation pattern very similar to that seen in both clinics and experiment. And we can identify ionic mechanisms responsible for this pattern and potentially, therefore, responsible for maintenance of AF. Come see poster 14. Thank you.
So next speaker, Tommaso.
Good morning. So I will present you in this poster the latest result we have obtained at INRIA and within the Healthy Child project about the personalized simulation of the cardiac function in tetralogy of Fallot and remodeling. So briefly, tetralogy of Fallot is a severe congenital heart disease, and the babies with that disease must undergo surgery very, very early, at 2 or 3 months. Unfortunately, that surgery may destroy completely or partially the pulmonary valves, which yields to chronic regurgitations And as the child grows, the right ventricle delays, and its function is impaired. The problem is that nowadays there is a technique where the clinicians propose to put new valves when the child is quite 15 years old, more or less. But as the lifespan of these valves are very short, they have to wait as long as possible. But if we wait too much, then the heart will be definitely damaged, and placing the valve won't be useful anymore. So the main question that we are trying to solve is, how can we first quantify the cardiac function in these children and of the right ventricle? For that we propose tools to calculate the blood pool volumes, ejection fraction from CNMR, but also to estimate the 3D myocardium strain from clinical data, from clinical MR. Then we would like to simulate the cardiac function of this patient, to simulate parameters that we cannot obtain directly in clinical routine, such as the pressure loops and so on. And for that we propose to use an electromechanical model that we personalize to these patients. And then we would like to predict what will be the shape and the function of these patients 1, 2, 3 years ahead, to help the clinician to decide if now is the right instant to put the valves or if he can wait a little bit more. And for that we propose a statistical model of remodeling of the right ventricle that we observe in a population of 18 patients. If you want to know more, come to see my poster number 15. Thanks.
Good. Next speaker, Pepijn van Horsen. Oh, am I wrong?
Wow, number 16.
Ah, Florence Billet. I saw, I didn't recognize you. Yeah, go ahead.
So good morning, everybody. I will speak about my poster. Will speak about the recovery of a cardiac motion and the estimation of the boundary condition by coupling a 3D electromechanical model and heterogeneous data. So on one side, we have a simplified 3D electromechanical model with cardiac phases included. This model is composed of two parts. One passive element, which is viscoelastic, linear, and anisotropic, and one active element, which is controlled by the electrical command U, which is directly related to the action potential. On the other side, we have heterogeneous data. We have 3D plus T medical images, such as CNMRI or CT images. And we can have electrical data related to these medical images, such as ECG or measured depolarization times. So we want to couple these two parts, the 3D electromechanical model, which is generic, with heterogeneous data, in order to personalize the motion of the model from this data. So we do it with a proactive deformable model. And in my poster, I will introduce the formulation of the implicit formulation of the image forces used in this deformable model, and I will show how using the 3D block matching algorithm in order to estimate the boundary condition of the heart. So the movie normally shows the results of this personalization, but I can show it near the poster this afternoon, and if you are interested, you can come and see the results and my poster.
Okay,
So
thank you very much. Okay, now comes Pepijn.
Good afternoon. I'm Pepijn van Horsen, a PhD student at the Biomedical Engineering and Physics Department at the Academic Medical Center in the Amsterdam, Netherlands. And my poster is about perfusion measurement and coronary vascular tree registration by means of an imaging cryomicrotome. Now, a cryomicrotome is a device capable of making high-resolution images of a whole organ. It consists of a large freezer with a cutting mechanism inside that can cut organs typically at 40 microns thickness. It has an optical setup with an excitation and emission filter wheel and a lens and a CCD camera. And the hearts are prepared by injection of fluorescent microspheres and fluorescent gas material, so that we can image the perfusion territories as well as the vasculature. And after cutting, we do some image restoration steps. We correct for any dark current due to the CCD camera. We correct for cutting artifacts due to the cutting mechanism. And finally, we do a deconvolution of the data by using a point spread function specific to the setup. Then we go to scent line extraction. We use a topology preserving thinning algorithm to get the scent lines of the vasculature. We classify the center points according to the distances to one another, so we can build segments and nodes, and from that we can do a radius estimation and look at the relative angles. And then finally we do the detection of the microspheres. We correct for spectral overlap, so we can extend the number of colors used to 5. We correct for transparency, and then we do a detection based on the morphological features. And here in number 5 you see the results. Left is the fringe representation of the vascular tree, showing separate benches. And on the right you see a cross-section of a heart with 2 microsphere distributions injected in the L6 and the LAD, and they can now be related to the segmental representation. I'd like to thank you, and I hope to see you at my poster.
Thank you very much. Next speaker, Jeroen van der Wijgaard.
Hello everyone, my name is Jeroen van der Wijgaard. I actually work at the Academic Medical Center. Let's remove this one. However, this work was...
Try yes.
Whatever. At the Academic Medical Center, however, this work was jointly done with a group of Nick Smith. This beautiful error message is overlying on the finite element model previously developed by Steve Nieder, which we actually wanted to expand by including a realistic representation of the arterial vascular tree. So for this we also used the imaging cryo-microtome. We took a porcine heart, filled the coronaries, sliced it up in 2,500 slices, stacked them back together, did some image analysis. That is the international symbol for it takes quite a while. We segmented out the ventricles and the epic heart. Then we ended up with the mathematical representation of the cylinders of the vascular tree, and that we recombined with the finite element model, which we sort of changed to represent our heart. So then we get a full integration of the vasculature into the finite element model. Well, the advantage of that is actually we can now look at the vessel deformation, at the angles and everything else, and also in the future we would like to include fiber orientation, things like that. So if you want to know more, please visit poster 18.
Okay, thank you very much. Next speaker is Jatta Rela. I think he has blocked this now completely. Take the next one quickly. Yeah, I know. Yeah.
Good morning, everybody. I'm sorry, I apologize for the movies. That's why there are blank patches on my presentation slide.
So that's a good reason to go to the poster.
So the main task of my work is to personalize the cardiac electrophysiology. By personalization, I mean estimating the parameters of the electrophysiology model so that the simulated depolarization and repolarization time isochrones are similar to the actual data. So the model we personalize is a Michel-Schaeffer model, which is derived from Fenton-Karma model, 3 variable. And we personalize to the data which is obtained using the DT MRI for the anatomy and local fiber orientation information, and optical data for the electrophysiology information. From that, we fuse these two data sets to get the depolarization and repolarization time isochrones on the actual mesh of the heart. And we also get the restitution property for each vertex of the node by using optical data, which is paced at different pacing frequencies. So now our task is to personalize the model so that we have the model simulated activation time and repolarization time isochrones and the restitution similar to the actual data. So the parameters which we personalize is conductivity parameter, APD parameter, and APD restitution parameters. So conductivity parameter is personalized for each zone in according to 26 zones for AHA, and APD parameter and APD restitution parameters are personalized locally. So you can see in the parameter distribution of APD the spatial heterogeneity of APD from base to apex. It's shorter in the base than the apex. So then we use it for ischemic stimulation. Please come to. You're welcome to my poster. Thank you very much.
Please come to the poster. Yeah, we'll just come poster is by Ayun Haid, but I hope you pronounce your name better than I do. That's the one? Good. Go ahead.
Good afternoon all, my name is Owen Hyde, I'm from the University of Oxford. I'd like to introduce some initial work on developing continuum model perfusion via the schematic. On the left hand side we have the general cardiac physiome idea of taking models into clinical benefit. Any mathematical model to be comprehensive will have to deal with blood flow in the microcirculation. We propose to do this perhaps through a continuum model where we take the tissue and assume it's a porous medium, where the permittivity tensors are derived using spatial homogenization techniques. They'll obviously have to be anisotropic. In parallel to this model, we also take the same data network and we solve for Purcell flow in order to compare these two. And the end goal, we take this into full-scale model. And if you want to see more, that's post number 20. Thank you very much.
Very good. David Northlet, go ahead.
Yeah, I'm Dave Northlet, and I'm also in Oxford with Nick Smith, and I've been doing my PhD on blood flow in the heart. We'll see if the movie plays. No. Excellent. Fabulous. All right. So anyways, what we've been doing is looking at coupling fluid mechanics inside the left ventricle with the ventricular wall mechanics. And we've tackled this from kind of a number of different levels. We first looked at the mathematical problem, and we set that up, and we did some analysis showing that the numerics and the mathematics are all set up in a way that's going to be functional for ventricular flow. Then we did some analysis, and what this movie doesn't show is the results of that, which are blood flow during diastolic filling. And we look to extend this into cases of whole heart function. We want to couple this to the left atrium and look at the kind of synergistic effects between the mechanics in the left atrium and the left ventricle, and then hopefully start to look at perfusion through the tissue and how that affects global function in the heart. So poster 21.
Thank you very much. Next speaker is Jack Lee.
Hello, my name is Jack Lee. I'm a newly starting postdoc at Oxford. I was mindful of the fact that if I delve into the details of my work, I'm going to need far more than 2 minutes. So instead, I thought I'd change the strategy and pull out the big picture stuff here. So here is a big picture. I have to thank Tim for granting me the permission to use it. I put this picture up here because I think it really well demonstrates how different microvascular networks can look in different kinds of tissues. And some of them have very clear structural organization, others have a very clearly identifiable units. And my main interest is in microvascular flow in coronaries because that is a site of functional significance. And when we first came to this field, we couldn't find any of these very detailed structural information in the literature. Fortunately, in the last few years, there has been very good progress. Progress is made in elucidating the myocardial microstructure. When you're looking at the microstructure of the coronaries, there's two things to keep in mind. As with other structures, topology is important, but I would also argue that the heart is not very useful if it's not beating. And in a beating heart, there's going to be different stresses and pressures acting and interacting with the flow here. So the spatial arrangement is another very important feature. And of course, this is complicated by the heterogeneity that exists within all sorts of different components, as well as how these two structures are coupled. So my poster used some of the high-resolution imaging work that I've done in conjunction with the group at Auckland. Some of the image processing and analysis techniques I've used to turn some 130,000 segments into graph-based description to allow analysis with respect to the myocardial damage structure. And somewhere in this great big mess, there is an organizing principle for spatial arrangement of the vessels, although you'll have to come and find out for yourself. So please come to see my poster out in the corridor where it's nice and cool. Thank you.
Okay, thank you very much.
Hi, So I'm Rebecca Shipley, I'm a mathematician over in Oxford, and the question that I'm interested in looking at is understanding how tissue scale, fluid and drug perfusion characteristics depend on the underlying structure. So the vasculature in tissues is a hierarchical structure, and one way to try and capture that mathematically is to divide it into, for example, arterioles, venules and capillaries. And then each of those different subsets is basically characterized by a different length scale. What that means mathematically is that you can use a process called mathematical homogenization to move between the different length scales. And what that lets you do is to derive the tissue scale perfusion equations based on the underlying structure. Now the process is flexible, so you don't actually have to prescribe the structure to derive the overall equations. And so that means that it's quite easy to test out different underlying structures and see what that means for perfusion. So that's kind of a brief summary, and basically on my poster I show a quantified case for that for vascular tumours. So that's basically it, and that's poster 23. Thank you.
Okay, thank you very much. Pablo Lamata.
Hello, good afternoon. My name is Pablo Lamata, and I work also in the computer lab in Oxford with Nick Smith. So my main motivation is trying to get all this research and try to move it to the hospital. I have more a bioengineering background, not in physiology. So I'm trying to push this, and in this poster I show a couple of contributions, a little contributions in this big challenge, which are the mesh personalization, trying to have the qubit Hermite meshes, which are the ones used for the biomechanical model in the COM lab, and try to make them personalized and try to guarantee that the quality of these meshes is enough to have robust and stable simulations. Not well. So for that we propose image processing pipeline in which at the end of the process we assess the quality of these meshes in terms of aspect ratio and orthogonality. And the second contribution is a little bit trying to understand what is the need of the cardiologist for cardiac resynchronization therapy, and try to convince them that the modeling methodology is trustful in the way that we believe in it, that the model is going to be a caricature of the reality, and the results we're going to predict are going to maybe help them or not in predicting the outcome of the therapy and making a better selection of patients. So in this user interface, we try to combine the classical parameters they look at in the heart with the model-driven parameters that we hope to get from the models. So poster 24, and let's go for lunch, I guess.
Thank you very much. Respectful to the audience, thinking of lunch. Very good. I think we've done a wonderful job. I mean, you all did a wonderful job. We're staying in time. And have a good lunch. See you in the afternoon at posters now. Not thinking, well, we now know it. I mean, now we go for the science. Thank you very much.